Zona pellucida. Good. Zona pellucida doesn't have any cells, and it has a lot of hyaluronic acid, which is the main chemical in connective tissue. There's a lot of hyaluronic acid in cartilage as well, so you can kind of think of this as a pretty good protective barrier surrounding that entire secondary oocyte. And we'll see that that can only be penetrated by a human sperm, and we'll go over how that works. And then there is a cellular layer around it, which is called corona radiata, and that is the cellular layer. Hopefully you remember we kind of saw how that developed within the ovary. We could see it starting in that secondary follicle. So this is what is ovulated. So now you have to imagine that structure is now entered into the uterine tube. The fimbria helped it. And now there is intercourse in that 3-day window so that the sperm can enter into the female tract. And again, you can kind of follow steps with the numbers. So here's a little sperm cell swimming up into the female tract. And the sperm will swim randomly. And because the sperm swim randomly, only a few of them actually get to the secondary oocyte. Of about 300 million that are ejaculated, only about 300 will get to that secondary oocyte, and you need that many in order to penetrate that zona pellucida, and we'll see how that works. And so sperm don't know where that secondary oocyte are, so they're just swimming completely randomly. And so just probability will have that some will get that far. And along their swim, there's going to be some functional changes to the sperm, and those functional changes are called capacitation. And so this is near the top of the handout, near the top of the uterine tube, and showing the capacitation. But what the capacitation is functional changes in the sperm. One of the changes is that they begin to be able to swim faster. So and these changes are due to fluids coming from the female tract. So fluids from the female reproductive tract will cause the sperm to be capacitated so that they can swim faster. And also what I was trying to show right here is that the membrane, plasma membrane of the sperm cell thins out a little bit. And I tried to make it, it's probably too small on the handout, but there's a thicker plasma membrane right here, and then due to capacitation, that plasma membrane is going to thin out a little bit. And so that is another thing that happens with capacitation. And this is again due to the female secretions, and as that sperm is swimming, it gets these functional changes called capacitation. And and that's going to, when you thin out the membrane so that that sperm cell can fertilize, or can have syngamy with that secondary oocyte, and then have fertilization occur. So number 5 on the steps is that the sperm will swim through the corona radiata. And so there's a little animation showing a sperm cell, and the cells there, the corona radiata. And then you have this picture right here. Where I put it in that handout is not where it normally occurs. You may remember normally the fertilization will be occurring in the ampulla, but it could happen near that infundibulum. And so that's what you see right here. The sperm, and there's several of them here, are getting through the corona radiata. And then right here you can see one sperm is now touching that zona pellucida. The zona pellucida inside has a protein called ZP3. And so only human sperm have a way to bind that protein. And so in the zona pellucida, there's this protein ZP3, and right here, that sperm cell is binding that ZP3. And when it does bind the ZP3, there's going to be enzymes released, and we'll see how that works. So I'm going to show an animation of what happens next. And just to orient you, here is a little cartoon picture showing the secondary oocyte right there that's blinking, and then what is surrounding that secondary oocyte is the zona pellucida. And so we see zona pellucida and then the secondary oocyte. And I'm going to zoom in to this zona pellucida, and so we're getting to high power, so we can now see this is secondary oocyte, and we see the zona pellucida. It's an acellular. You have to think of it as kind of like hard cartilage, or soft cartilage or hard connective tissue. It's kind of the consistency of it. Protective barrier. And then these little proteins in the zona pellucida are the ZP3. So you can see the ZP3 is just all over in the zona pellucida. And so it's surrounded also by hyaluronic acid, some of the chemicals that would be in the connective tissues. So now we'll look at a sperm, and this is the head of a sperm cell. And on the plasma membrane in the front, there is a protein that can bind onto that ZP3. And so humans have this protein, other species wouldn't have this protein. So this protein can bind onto that ZP3, and so it's species specific. Also within the head of the sperm cell, there is an organelle called an acrosome. That acrosome has enzymes inside that can break through the zona pellucida. And so this is an important organelle with lots of enzymes. One of them is hyaluronidase, which can break down hyaluronic acid. Also in the head of the sperm cell is a nucleus, and that would contain the haploid number of chromosomes. And then there's something called the midpiece, and this is in the sperm where the mitochondria are. The sperm has to swim a long distance, and so you need a lot of energy, or the sperm needs a lot of energy, and so the ATP would be made right there in the mitochondria. And then you can see the tail right there. And we'll look at the sperm in the microscope later in lab as well. And so right there again is that protein on the head of the sperm that can bind the ZP3. And right there is the acrosome that contains enzymes that can break through the zona pellucida. And so now we're going to look at this acrosomal reaction. And the sperm is swimming, and notice that protein now binds the ZP3, and when it does, you'll have this acrosomal reaction, which is where the enzymes from the acrosome will be released, and those enzymes can break through the zona pellucida. And so the enzymes are breaking through the zona pellucida so that the sperm cell can swim and get to the secondary oocyte. This is syngamy, where that sperm plasma membrane binds onto the secondary oocyte plasma membrane. You may remember syngamy from that first lecture. And so one thing to recognize on that animation, it wasn't realistic in the sense that one sperm went all the way through the zona pellucida. That actually doesn't happen. More realistically, you have the enzymes here, and they will be released through the acrosomal reaction, and maybe only this much will be broken down. And so you're in a sense just taking one sperm can only take one little bite out of that zona pellucida. And so after this one has cut a little bit into that zona pellucida, there's going to be one behind it that will go a little further. And that's why you need at least, they say 300 sperm in order to get all the way through the zona pellucida. And that's why if a male, even has 80 million sperm in an ejaculation, that's probably not enough to get through that zona pellucida. And so, but this is the acrosomal reaction. The enzymes from the acrosome are going to break through the zona pellucida. And it's due to that protein binding onto the ZP3. And you can see how there's ZP3 all the way through, so that the next sperm is going to have a ZP3 it can bind onto. And so that is the acrosomal reaction. End result is we have syngamy. And so syngamy is the fusion of the plasma membrane of the secondary oocyte and the sperm cell of the two gametes. And so this is an animation showing syngamy, the sperm cell's nucleus fusing with that secondary oocyte nucleus. And this is going to cause several things to occur. But first thing we'll see is that we have a model showing syngamy. And so now you can see on your handout, and this top one right here. And so this one right here is the one you have on the handout that looks like this. This one is showing syngamy. And so in the lab exam, you could be asked, and it could be pointed right there, what process is occurring right here? And your answer is syngamy. We are looking at the head of the sperm cell touching the plasma membrane of the secondary oocyte. So that is syngamy. And with the syngamy, there are several things that it will cause. And so we'll go through those. You could also be asked that in the lab exam. This is in the handout. And so one thing that the syngamy causes is a fast block to polyspermy. Polyspermy just means many sperm. And so what we're doing is we're blocking syngamy, or blocking other sperms from contacting the secondary oocyte. So you're just blocking other sperm from touching the secondary oocyte. Which should make sense. For a baby to happen, you just need one sperm cell. You actually don't want several sperm to put their nucleus inside the secondary oocyte. You just want one sperm cell nucleus. And therefore, once you get one sperm to touch that secondary oocyte, you want to block all other sperm. And that's what you're having with this block to polyspermy. And so there's going to be a fast block and a slow block. The fast block is a depolarization of the secondary oocyte. That's what this animation is showing you. Notice right when the sperm touches the secondary oocyte, they're going to be showing a depolarization. And it just stopped when, I think you saw it. For some reason it just stopped when I had said it. And so when this sperm cell touches that secondary oocyte, right there you see the depolarization. And so because you get depolarization around the entire secondary oocyte, a sperm, even on the other side, won't be able to cause syngamy. Only one sperm does, and instantaneously, once one does, you get this depolarization preventing other sperm from contacting the secondary oocyte. So the idea would be, here you can see the sperm, and you'll see one is going to touch the secondary oocyte, and when it does, there's going to be this depolarization, a literal electric shock that occurs. And when that happens, these other sperm will, oh, bummer, I didn't get it. It's just when that shock happens, it's done. One of them got to that secondary oocyte, the other sperm won't be allowed in. And so that is the fast block, this depolarization that can occur. Slow block is where that zona pellucida, the remains of the zona pellucida, will actually calcify. Let's say that this is the secondary oocyte, and then the zona pellucida will actually begin to calcify. And so it's actually starts from the depolarization. The depolarization that occurs, which is the fast block, is going to cause the zona pellucida to calcify. When the zona pellucida calcifies, it becomes like a hard shell. Just like a chicken egg has a hard shell, now this zona pellucida is going to have a hard shell. And so now you have this slow block, we call it, because with a hardened zona pellucida, there's no way any sperm can get in there, because now it is calcified. And so that is the slow block, calcification of the zona pellucida. So another thing that the syngamy causes is the nucleus of the sperm cell will be injected into the secondary oocyte. When that nucleus goes in, it will become the sperm cell pronucleus, you may remember. And so that is going to happen after syngamy. And one last thing that happens because of syngamy, you may remember, is that syngamy is going to cause meiosis II to resume. So that secondary oocyte was paused right here in metaphase II, but when you get syngamy, now you're going to have anaphase II, and then you'd have telophase II, and the end result is going to be the ovum and that second polar body. And so we have right here a picture of what the ovum would look like. If this cell, which actually I have it in black and white here because this is not in this model. So the ones that are in color are on this model, but this is one that I just made so that you get the idea of what the ovum looked like. I showed this, I kind of drew a picture of this in that first lecture. But an ovum would be a cell that would have two pronuclei. One of the pronuclei would be the pronucleus of the sperm cell. The other would be the pronucleus of the ovum. And so the ovum technically wouldn't be around too much because soon after the ovum is there, you're going to have fertilization. And this is a colored picture, so we do have this as a model. And so this is the second one in the middle here. We had syngamy on the top, and now we have this one that you can see right there in the middle. And so you could be asked, what is the process shown right here? And the answer is fertilization. And so this is where you have those pronuclei fusing. And so that is fertilization. And so this model shows a good picture of fertilization. And as a result of fertilization, you have the zygote. And so we do have a zygote in this model as well. The bottom one there, and you have it in the handout, is the zygote. So this is one cell, and this is the zygote. They are showing right here the first mitotic division. And so what we're looking at here is mitosis. And so once you get the zygote will immediately start to divide by mitosis. And but that is the zygote. So you can see right here. The model's accurately showing it in that zona pellucida. So we still have zona pellucida. Right now this zona pellucida is calcified because of that slow block. So this would be a very hard and shell-like zona pellucida and helping to protect this zygote. And you can see the two polar bodies. So now after the zygote forms, so we just went from fertilization to, from ovulation to fertilization, now we'll go from, to implantation. We'll be ending with the implantation. But this is the zygote, and again, we have a cell showing the zygote, and the zygote will start to divide by mitosis. And so this one zygote will become 2 cells will become 4 cells, etc., and that's what we are looking at right here. And so this model is showing this process called cleavage. So cleavage is this rapid mitotic divisions of the zygote initially. And as you can see, the zygote right here, then 2 cells, then more cells, more cells. The process is called cleavage. And so we have that on this model. And so I could have a little piece of tape going across the model and saying identify the process. This is cleavage, just rapid mitotic divisions occurring all within the zona pellucida. And so when you see this, it's pretty rapid, so there's no time for the cells to grow. So as the cells divide, the cells themselves are becoming smaller and smaller and smaller because they are all still in that same hardened zona pellucida, this calcified zona pellucida. The end result of cleavage is a ball of cells, and we have this on the model as well. And so the ball of cells right there is called a morula. And so this is the entire structure you would have right here. Result of the cleavage is the morula. And the morula would still be in that hardened zona pellucida. Each one of the cells there is called blastomeres. And as we're labeling this, these are all on the list as far as the lab is concerned. So these are things that you need to identify for lab. So the cells, each of the cells is a blastomere. So you get the idea that this moments before here was a zygote, but once it divided, then you have 2 blastomeres. The blastomeres divide, become more and more blastomeres until you have a ball of blastomeres called the morula. Here is a view of a real zygote becoming 2 blastomeres. Notice in this video, this is the hardened zona pellucida. You have the zygote right here, and then you have a little polar body right there. And then we can see at least the first mitotic division. And so this is the zygote. It undergoes mitosis, and now we have 2 blastomeres. And then these blastomeres will divide by mitosis and become more blastomeres until there's a ball of these blastomeres. And all within the zona pellucida. So this is happening around days 1 to 3, and the end result is the morula. One thing, and I'll talk about this next time, is when there are identical twins, or identical twins happen when these blastomeres separate. And so each one of these blastomeres, if you separate them, there would be separate individuals. But because they came from the same zygote, they would have the same genes. And so identical twins would be where you have these blastomeres separating early on, and then you'd have identical twins because they came from the same zygote. So they'd be monozygotic twins. That's in the handout as well, and we'll talk more about that on Tuesday. So this is the cleavage where you have the zygote dividing rapidly, and the end result of the rapid mitosis is the morula. The morula is the structure that actually enters into the uterine cavity. And so right here is a morula. So when you're looking at this, you have to imagine it is a ball of cells. It is then going to move and go into the uterine cavity. And so remember, right here you have a nice fresh stratum functionalis. And the stratum functionalis right here would be in that secretory phase, so there's a lot of glands in the stratum functionalis, and those glands will be secreting out a fluid, and they call it uterine milk. This fluid actually will go into and penetrate that zona pellucida and go into the morula, and it's going to cause a fluid-filled space within the morula. And so that's what they're going to show right here. Here's the morula. And now you have to imagine it has entered the uterine cavity, and then the fluid secretions from the uterine cavity is going to enter into the morula, and we get what's called a blastocyst. And so now the morula has changed its name to a blastocyst, and on this model we actually have 2 blastocysts, one small one and one large one. And so they're not showing on the model the zona pellucida, but I think this one is about the size of the morula, so you have to imagine that this one probably has a zona pellucida around it, and then this one will be after it leaves the zona pellucida. But both of those are just blastocysts. You don't need to know big or large. They're just two examples of this blastocyst. A cyst is just a structure that has fluid in it. It's a fluid-filled sac. So what we have here is fluid, and so that's why they call it a blastocyst. Still within that zona pellucida. Next thing that happens is that the blastocyst, it's in the uterine cavity, it's going to literally hatch from that hardened zona pellucida. Remember, the zona pellucida was calcified, and so now you have that blastocyst literally hatching from the zona pellucida. Just like a chicken would hatch from its egg, now that blastocyst is hatching from that hardened zona pellucida, and this needs to happen so that implantation can happen. And so the blastocyst is going to leave in the uterine cavity the zona pellucida. And there's little enzymes that it would secrete to cause it to be able to break through the zona pellucida. And once it has left the zona pellucida, it would then be a little bit bigger. And then we have, so you can see, again, two blastocysts. One is bigger than the other. And that's why I like to think of this one as the one that is hatched, obviously, from the zona pellucida. And so it is now this blastocyst that can implant into the endometrium around day 6. One thing though, and this is where you might want to just take note, the blastocyst has two types of cells. The cells right here, little kind of a mass of cells, these cells are called the inner cell mass. And so this is where we start to have differentiation. So those cells right there called the inner cell mass are the cells that eventually will become the embryo. And so they are, and we'll see how that works a little next time. And so they are the inner cell mass, those cells right there. The cells that are on the outside of the blastocyst are called trophoblasts. And so the trophoblasts are going to be these cells on the outside. Just so it's very clear, if this were on the lab exam, I'd definitely point to the outside here rather than the outside there, so it's clear, you know, that this would be trophoblast, and right there would be the inner cell mass. Trophoblast has a very important function in that it secretes the HCG. And so the trophoblast right here, these cells secrete that HCG. So very early on, you get the secretion of the pregnancy hormone, HCG, from that trophoblast. And hopefully you know what HCG does. It rescues what? The corpus luteum. So HCG is going to rescue the corpus luteum. Remember, normally during the female reproductive cycle, you have lowering levels of LH. And if the LH gets too low, then the corpus luteum would die. But now, because you have HCG, it's going to prevent the corpus luteum from dying. And as long as the corpus luteum is alive, it will keep secreting estrogen and progesterone so that the endometrium will not be shed. Obviously, that blastocyst did not want menstruation, and so it's going to be saving. First thing it's going to do is rescue that corpus luteum so that it's home right here, the inside of the endometrium, or the endometrium will still be there. And so the next step would be implantation. This is just an animation. This would be the stratum functionalis, and then this is the blastocyst. And what the blastocyst is going to do is just find a place there on the endometrium where it can burrow itself in. And this is just an animation showing what the implantation would look like. And so it's going to release some enzymes, and when it does, it literally will burrow itself in, very much like a parasite does. And so there's just enzymes that will allow it to burrow in, and then it finds its home within that stratum functionalis where it will start to develop. And so this is the implantation. And so just to kind of review it all, this is an animation going from ovulation to implantation. You can see the uterus, you can see the ovaries. And so we'll start with the ovulation. So the ovulation is of that secondary oocyte. It would be paused, as you know, in metaphase 2. The fimbria will bring that secondary oocyte into the uterine tube, and then there will be sexual intercourse that will allow the sperm to enter the female tract. They'll go up to the uterine tube. Around the ampulla is where the sperm will then contact the secondary oocyte. You'll have syngamy, and the syngamy, as you know, is going to cause the meiosis II to resume, and then that secondary oocyte will become an ovum. The sperm cell pronucleus will merge with the ovum pronucleus, and then you have the fertilization. Now you have the cleavage, where you have rapid mitotic division until you get a morula. The morula will enter into the uterine tube, secretions will enter into the morula, and you get a blastocyst. The blastocyst will then hatch from that hardened zona pellucida, and then the blastocyst will be able to implant into that stratum functionalis. And so that would be the implantation. And so then on Tuesday, we will go on from there and just, in a summary form, not go over all the details of embryology, but get the big picture of how this little blastocyst will then develop into, a embryo, fetus, and then a baby.